All right. This is now being live streamed to pick up those handful of folks that like to join us through those channels. So I'm going to go ahead and close these polls. And thanks again for those of you who are members of New Hampshire Audubon, and a special welcome to those of you who are not yet members of New Hampshire Audubon. It's great to be here with you all this evening for an awesome talk. So, you might be asking yourself, who is this guy? My name is Mark. I'm the Conservation Program Director here at New Hampshire Audubon. And I would like to first acknowledge that this presentation is streaming to you from our state headquarters in Concord, New Hampshire, which is located within the ancient site of Pennacook in the Nadakina, which is the traditional ancestral homeland and waterways of the Abenaki, Pennacook, and Wabanaki peoples past and present. I would like to acknowledge and honor with gratitude the land and waterways and our ancestors, the Alnumbak, or human beings, who have stewarded Nadakina throughout the generations for thousands of years. New Hampshire Audubon is honored to continue this stewardship of these lands, providing opportunities for all people to form connections to the natural world through our programs and wildlife sanctuaries around the state. And I invite you to learn more about the Indigenous presence on the land you occupy by visiting the website native-land.ca. Here you can explore and click on territories of Indigenous people and get connected to resources to learn more. And for a more in-depth understanding of the Granite State, check out all the educational resources at indigenousnh.com, including this interactive story map that details the Indigenous presence and their stories here in New Hampshire. And thank you again for your interest in tonight's topic, Motus, a revolution in migration research, presented by our own Dr. Carol Foss, who is the Senior Advisor for Science and Policy here at New Hampshire Audubon. I have the privilege of working with Carol on a regular basis, proposing grants that support her research, as well as implementing the large-scale Motus array here in New England. I always learn something new from Carol, so I'm really looking forward to hearing her talk alongside you this evening. And as you may know, this talk is the 19th session of a year-long webinar series called Exploring Connections to and Stewardship of the Natural World, sponsored in part by a grant from the New Hampshire Humanities Council. The past recordings of these talks can be found on the New Hampshire Audubon YouTube page, as well as the series webpage where you found this registration to get here. And throughout this series, as you may know, we're exploring that intersection of the sciences and the humanities, finding and forging new ways to connect with nature and learn about the importance of conservation action. I'd like to invite you to really take the time and space to consider how tonight's topic informs, strengthens, or otherwise supports how you define yourself as a person and how you can use this topic to connect with human communities as well as the wild ones. Before I hand it over to Diane to introduce Carol, I'd like to take this opportunity to briefly describe how this webinar fits into the larger mission of New Hampshire Audubon. For those of you who don't know, New Hampshire Audubon is a state-based environmental nonprofit that's completely independent from National Audubon. We rely on members and donors like you to support our charitable mission, which has 4 programmatic pillars: connecting to people, to nature through environmental education experiences like school programs, nature day camp, and webinars like these; researching and conserving species in peril, including large raptors and small birds; managing about 10,000 acres of wildlife sanctuaries throughout the state for habitat and recreation; and finally, advocating sound environmental policy in the New Hampshire State Legislature. I'm here today because of members and donors like you. We also rely on a huge network of volunteers that assist us with wildlife monitoring, ambassador animal care, environmental education, and wildlife sanctuary management throughout the state. If you're a volunteer, member, or supporter of New Hampshire Audubon, I would like to sincerely thank you. We simply couldn't achieve our charitable mission without you. And if you'd like to become a part of our conservation family, which I hope you will, please check out our website for ways to get involved. We do have over 90 people registered for this evening's talk. So you'll see that we're in full webinar mode. So we're going to use the chat for any thoughts, comments, and reactions you might have, and reserve the Q&A button for any questions that you'd like specifically answered by Carol. It's been great seeing how far we've been able to reach through these webinars. So if you are willing, go ahead and type into the chat where you're watching this presentation from. And I want to give a quick shout out to Diane DeLuca, who's the senior biologist responsible for orchestrating this series. Without her leadership and coordination, this webinar series just would not happen. So thank you, Diane. And speaking of Diane, she and I will be monitoring the questions both here in Zoom as well as Facebook Live throughout the evening. And we'll feed those questions over to Carol at the end of her presentation. So without further ado, I'd like to hand it over to Diane to introduce Carol.
Thanks, Mark. And I always like to give a shout out back to Mark, because without Mark, this webinar series wouldn't happen anyway. So thank you, Mark, for making this something that we can actually do. So Dr. Carol Foss is the Senior Advisor for Science and Policy at New Hampshire Audubon. And she has served New Hampshire Audubon in a variety of capacities for more than 40 years. I believe the longest tenure of anyone at this organization. She began her career as the resident naturalist at the Paradise Point Nature Center during the summer of 1975. Following that, she joined the headquarters staff as part-time education director in 1977, and soon shifted to director of wildlife programs during the time when New Hampshire Audubon partnered with the New Hampshire Fish and Game Department to develop the New Hampshire Endangered Species Program. She currently holds the position of Senior Advisor for Science and Policy. Carol holds a bachelor's degree in biology from Colby College, a master's degree in zoology from the University of Connecticut, and a PhD in wildlife ecology from the University of Maine. Carol is a native of New Hampshire and lives in Pennacook, where her ancestors settled in the 1700s. She is especially interested in bird behavior during the breeding cycle and in the influence of human activities on how birds use the landscape. Her favorite places to do fieldwork are the Androscoggin-Megalloway watershed in northern New Hampshire and western Maine, and in northeastern Peru. And tonight, as Mark already mentioned, Carol is going to be sharing an introduction to the Motus Wildlife Tracking System, which is a new research network that is revolutionizing the study of winged migration by tracking the movements of small birds, bats, and even dragonflies and monarch butterflies who have been fitted with tiny radio transmitters called nanotags. Carol will discuss how Motus works, what we're learning from it, and efforts that are underway to expand the network in the Northeast. So thanks, Carol. We're really excited to have you tonight.
This is the first time I've done one of these with two screens, so you've got to bear with me. And fortunately, Mark is right here to come to my rescue. So thank you all for being with us tonight. This is a great crowd, clearly more folks than we would have had if we had done this in person here. So thanks all for being with us. In order to appreciate how much of a revolution Motus really is, I'm going to start with a quick review of the tools we had for tracking birds before Motus became available, then proceed to an explanation of Motus, describe the efforts underway to expand the network in the Northeastern United States, and then, if you're still with me, end by sharing the travels of some birds that have been tagged in New Hampshire and some that have migrated through the state in recent days. Traditional radio telemetry, which has been around since the 1950s, involves use of VHF, very high frequency radio transmitters, each of which has its own unique frequency. Biologists use scanning receivers to detect individuals and then use directional antennas to home in on their locations. This is all well and good when the tagged individuals remain in a relatively small area, but presents challenges when they move further afield. We drive available roads with omnidirectional antennas attached to the roof and extend the antennas on a pole for lost birds. And it's even better if we can get to the top of a hill to do that. When things get really desperate, we sometimes resort to an airplane, which gives us about a 2-mile detection range compared to about half a mile on the ground. And airplanes obviously can also cover a lot more ground than a truck can in a short time. But in any event, using this tracking technology to track migration is definitely out of the question. In the 1980s, satellite transmitters were developed that can be used to track large birds and mammals, but they are much too heavy to use on songbirds. Use of cell phone technology soon followed to transmit data directly to researchers, but weight limitations still kept songbird biologists from using them. The next innovation was the development of light level geolocators, which were originally used, originally used to track marine invertebrates, and were first used on birds in 2007. And these devices have light detectors that sense sunrise and sunset, and then from those values, complex formulas can estimate the bird's location to within approximately 200 kilometers, which is about 124 miles. This was the first technology that was lightweight enough to use on songbirds, but the rub is that you have to recapture the tagged bird the following year and remove the device in order to download the data and do all the complex calculations. While the resulting data is not very precise, this technology did provide the first broad-scale data on migratory connectivity of songbirds. We had those for a few years, and then pinpoint geolocators became available a few years later. Their advantage is they give you precise GPS locations for programmed dates and times, but they also have to be downloaded, recaptured and downloaded, and they only can hold a limited number of locations. So while the light level geolocators give you continuous data for the whole migration, the pinpoints only give you, well, the ones we used gave us 50 locations. So you can't have everything. But then came Motus. And Motus is a collaborative global research network to track wildlife movement. Motus is not an acronym, it's the Latin word for movement. And this system uses automated radio telemetry with digital radio tags that are all on the same frequency. And being all on the same frequency, you can install receiving stations all over the landscape, and they will detect any tag that passes within about 15 kilometers line of sight. So you don't have to recapture the animals, which is huge when you're dealing with birds that are migrating over long distances. And even if the bird does not survive to come back to its breeding area, you still have an opportunity to get some data from it. Tags are made in a range of sizes, and can be installed on birds, bats, and as Diane mentioned, even large insects such as monarchs and green darners. The Motus network was originally set up to track migration of shorebirds. So the first receiving stations that were installed were all along the Atlantic and Gulf coasts and along the shores of the Great Lakes. Some of the early research subjects included semi-palmated and white-rumped sandpipers, dunlins, piping plovers, and the endangered red knot. As some folks may be aware, Delaware Bay is a very important migration stopover area for red knots. They feed there on the eggs of horseshoe crabs and migrate north to their breeding areas from there. So that was a major tagging area for red knots. What was discovered from that effort was that the majority of knots that forage in Delaware Bay then cross Pennsylvania on their way to their breeding areas in Arctic Canada. Now, red knots are globally endangered. They're a shorebird. Pennsylvania has a very tiny shoreline. So it never occurred to anyone to put red knot on their threatened and endangered species list. However, discovering that a substantial part of the population migrates over Pennsylvania, this data enabled the state to list red knots, and they use this data to help inform locations of wind farms on the ridgelines in the state. And Pennsylvania has lots of wind farms. So this is one application of Motus that has had a direct conservation impact. So once this network of stations was set up, folks studying salt marsh sparrows, which are also a species in trouble on the East Coast, realized that they could use the network to enhance their research. So that was the first songbird application of Motus. Then in the mid-teens, a far-sighted, forward-thinking group of biologists in the mid-Atlantic region recognized the potential for studying forest songbirds using Motus and decided to work towards creating an inland network of receiving stations in this black hole that is the Northeast. So we had a regional meeting here at the McLean Center in 2017, I think, with biologists from all over the region who were interested in this goal of expanding the network within the Northeast. And the group decided to pursue an application to the Fish and Wildlife Service for a competitive state wildlife grant, which is informally known as CSWIG. The idea was that they would move forward with an application for the mid-Atlantic states first. And if that was successful, then we would follow the next year with a proposal for expanding the network in the New England states. So the Mid-Atlantic proposal was indeed awarded. It's a 3-year project from 2019, and it's finishing up this year. It funded receiving station installations. I think there were 45 in Pennsylvania, Maryland, New Jersey, Delaware, and New York, and field projects on 8 species of greatest conservation need in the region. And we were fortunate enough to have our Rusty Blackbird research selected as one of the 8 field projects in that initial C-SWIG. So in 2019, we tagged 9 adults. 8 of them were detected during fall migration. 2 were detected on their way back north the following spring. And 6 of them returned to the breeding grounds, including one that was never detected during either spring or fall migration. So that bird clearly took a different route than everybody else. And this is a map of where those 8 birds were detected in their travels. You'll see that a couple went as far as South Carolina, and then one went all the way to Georgia. We kind of lost a bunch of them around Chesapeake Bay, but we suspect that those birds wintered inland in the southeastern states, which is kind of the current black hole in the east. We were also able to install a small receiving station in Dixville within our Rusty Blackbird breeding study area under the Mid-Atlantic Grant. So in 2018, after the successful acceptance of the Mid-Atlantic Grant, we started working on a C-SWIG application for New England. That was approved for 2020 to 2023. It's close to $1 million with a 26% match in-kind services and donations. New Hampshire Fish and Game is the lead agency. The Maine, Massachusetts, and Pennsylvania state wildlife agencies are partners. And partnering non-governmental organizations include the Carnegie Museum of Natural History, Maine, Massachusetts, and New Hampshire Audubons, and the Willistown Conservation Trust in Pennsylvania. The first objective of the New England project is to install 50 inland receiving stations within the region. And by siting these stations in what we refer to as fence lines, with the receiving stations about 30 kilometers apart. The idea is that any tagged individual passing through that fence line should get detected by at least one receiving station. So this was kind of conceptual fence lines, and they never turn out quite as straight as your concepts, but we do the best we can. So the current status, despite kind of a slow start with COVID interruptions, we've investigated 5 potential sites in Connecticut. One has been installed. There are actually 3 scheduled now. We've investigated 33 sites in Maine and have now installed 4. 39 in Massachusetts, and 3 have been installed and 2 existing stations upgraded. So you can get the idea from this slide that we have to investigate a lot of possibilities before we actually settle on a site. And that process begins with kind of looking at maps and looking at heights of land along those fence lines and looking for potentially interested landowners and then honing in on a few sites and contacting potential landowners. So it's a long process before you get to the actual installations. So this map shows where the current receiving stations are. And I'm going to stop here and take any questions that may have arisen so far, because the next stretch of the presentation will be visiting the New Hampshire receiving stations and exploring the travels of some of the birds that have been detected at them. So this is the time for technical questions about the project overall and Motus overall.
Oh, here's a couple. Was the program initiated in Canada? There are so many Motus stations in the Maritimes in Southern Ontario versus the US.
Yes, the whole Motus network is a project of Birds Canada, and started up there. I should have mentioned that. So thank you for the question.
Someone's wondering, what are the weight ranges of the Motus tags?
Oh man, the smallest one is, what did we put on the monarchs, Diane? 0.13
0.13.
of a gram.
0.13 grams.
Yeah, so those are the tiniest ones. The ones that we use on rusty blackbirds are just about a gram. The larger the tag, the larger the battery is in it and the longer it lasts. So we can get 2 migrations out of the 1 gram ones. There are also larger ones that they put on woodcock and some of the larger birds, and I don't know how much those weigh. Probably 2 to 3 grams, I would guess.
And Carol, we might just mention that this was the first year that they actually produced the 0.13 grams
Right.
nanotag, which they are calling a nano pin. Prior to that, any of the insects were outfitted with a 0.23 to 0.27 gram nanotag, which is still a substantial weight for an insect or a monarch.
Yes.
So someone was wondering what you mean by an upgrade.
Okay. An upgrade of a station generally involves putting up more antennas, a more substantial mast to support the antennas, and there's a whole new set of tags that I'm not going to talk about tonight called life tags, which are powered by tiny solar panels, so that they can potentially last for the entire life of the bird. They operate on a different frequency from nanotags. So the upgrades are also getting equipment that detects the life tags. So that means 4 more antennas and a second receiver. So that's what the upgrades involve.
Carol, a couple more. The price for tags?
Yep.
Someone's wondering what the price for the tags are.
Yeah. We're paying about $250 for the rusty blackbird tags. Do you remember what we paid?
Yes, we pay. So the nano pins are the most expensive. They're about $275 each. But if you get a slightly larger nano tag, I think it was $175 each.
Yeah, it kind of depends on the size of the tag and how many biologists are using that size. So the more they can mass produce, the prices are a little lower. But COVID has kind of messed things up because the supply chain for some of the components of these tags has been really delayed, and we had hoped to put out, I think, 15 pins this year, and we couldn't get them because of supply chain issues. So we're hoping that will get straightened out before next fall. Another question?
Yeah, here's one about the base stations. How are these base stations configured? Solar power, Raspberry Pi computer-based system, connected via BMC to antenna? Curious how this is done on the technical level.
Give me a couple more minutes to get to one of these stations, and I'll talk some more about that.
Okay. How are the tags
Okay.
put on? How are the tags put on?
Okay, it depends on the species and the purpose of the study. With the monarchs, we glued them on. There's not really any way to attach a harness to an insect. With the rusty blackbirds, we use a leg loop harness, and the tag sits on the back of the bird like a backpack. I think some of the shorebird ones, they may be attaching to legs. I haven't been directly involved with any of that research, so I don't know. But basically, it's usually either glue or some type of a harness.
And Carol, last question here. I have trouble understanding how you tell individual birds if they're all on the same frequency.
That was a real mind blower for me as well. I had a hard time wrapping my mind around it. But what it boils down to is each of these transmitters pulses a unique signal. So it might be sort of like Morse code, although I've been told that isn't a good comparison, but basically it's a burst frequency. So you can order different burst frequencies, and then each individual has a unique pattern of transmission that's repeated every, I think we're doing 9 seconds. Some of them are shorter, some of them are like 20 seconds. And the longer your burst interval, the longer the batteries will last. But if you're trying to track any of these birds by hand before they leave on migration, we've been able to do pretty well with the 9-second burst interval, but a 20-second burst interval would be really, really hard because you'd have to just wait for the next burst to get a bearing on where the bird was. Anything else for now?
That's it for now.
Okay, well, don't hesitate if you have additional questions that occur to you. Feel free to pop them into the Q&A and we'll tackle them at the end. So the configurations of these stations differ from one site to another, but this is kind of the basic pattern that we're using in the Northeast. We use 2 sections of roan tower, which are those towers you see with equipment on all along the interstates these days. If it's in a remote site, there's a solar panel to energize the receiver. There's a big waterproof box that holds the receivers, the connections to the antenna, the batteries, and kind of everything that connects to everything else. We were using Raspberry Pis initially, These stations that are being upgraded had Raspberry Pis, but now there is a receiver being manufactured called a sensor gnome, which will detect both nanotags and life tags. So upgrades are going to those receivers where you kind of get 2 for 1. So I'm going to start with our northernmost station, which is on Crystal Mountain in Dix's Grant. If some of you saw the recent Audubon publication, we put in a whole sequence of photographs showing the installation of this station, which took all day, which is why the light is a little dim for this picture. Crystal Mountain is located about halfway between routes 145 and 116, north of Route 26 from Errol to Colebrook, if any of you are familiar with the North Country. And let's see here. One of the first birds that was detected at Crystal Mountain, other than our local rusty blackbirds, which I have not tried to include in this presentation. There's a bird observatory on the north shore of the St. Lawrence River, the Tatusac Bird Observatory. And they have a big project going this fall tagging American pipits and horned larks. And they're still tagging, so they don't have all their date and registration information into the website yet. So we're not sure exactly when this bird was tagged in Quebec, but it was picked up at our Crystal Mountain site in Dix's Grant and also at our station in Berlin on the 19th of September. And then by the 11th of October, it had made it all the way to the Gulf Coast of Florida. So without triggering any detections anywhere else. So that's kind of interesting. Another bird from that project, and I'm not as convinced that it was a pipit because it hasn't gone as far, was tagged in Quebec, traveled south along the St. Lawrence, then triggered 2 of our stations in northern New Hampshire, crossed over to Shelburne, Vermont, and then headed down towards Albany. So we'll see where that bird goes in the coming weeks and if it gets detected again. But one of the things you can do on the Motus website is click on those white dots and it will tell you the name of that receiving station. And then you can go down a rabbit hole if you want to and find out who's sponsoring that station and what else has been detected there. You can also click on those white lines between stations. And if a bird was detected by 2 sequential stations on the same day, you can get an estimate of the speed that it was traveling. So this bird had estimates on different legs of its journey ranging from 22 to 71 miles an hour with a mean of 39. And so, I mean, sometimes they're going to have headwinds, or sometimes they're going to have tailwinds. Sometimes they may stop to forage. So the speeds are going to vary, but it's pretty interesting to look at those estimated flight speeds. There was also a hermit thrush that was tagged in New Brunswick, detected at 3 of our northern New Hampshire stations, and spent 21 hours in Berlin. So since these are night migrants, it's probably spent a day foraging within detection distance of our Berlin receiving station. and then headed down and was detected in northeastern Connecticut and on Long Island. And hopefully we'll hear from that bird again in the coming weeks. So our South Valley receiving station, as you can see, this does not look quite as substantial as the Crystal Mountain Station. This is what we call a pop-up, and it just uses a monopole mast rather than a whole roam tower for supporting the antennas. You'll also see that this was installed before we had the life tag and sensor stations. So this only has 4 antennas instead of 8. We deactivated it last winter because they had had 13 feet of snow locally the previous winter. And we were a little concerned about running the battery way down if the snow got up over the solar panel. So we just brought the battery in, wrapped the solar panel with a lot of padding and a waterproof tarp to protect it from ice, and just trickle charged the battery all winter. It was reactivated in April, and the South Valley Station is located not too far north of Route 26, again, between Errol and Colebrook. So I'm including some of last year's detections there because they're pretty interesting, and the Canadian Wildlife Service was still pretty much shut down this year because of COVID, so there weren't very many shorebirds tagged. But this is one of those red knots that was tagged on the South Atlantic coast between Beaufort and Charleston, or North Carolina, South Carolina. I didn't fix that. And then it moved north of Charleston and then made a beeline north and was detected in Pennsylvania, then up to the shore of Lake Erie, and then went offline for the breeding season. Then in the fall, the first place it was detected was at our South Valley Station, and then it wasn't detected again until it got back to the same area where it had been tagged in the spring. Another red knot was tagged during stopover in Delaware Bay, and was detected passing through eastern Pennsylvania at a whole series of receiving stations on the 1st of June. By the next day, it was on the west shore of James Bay in Ontario. And then in the fall, passed over Dixville Notch. So I think some of the shorebird biologists have been surprised that their birds have been coming as far east on their fall migrations. But I think some of them may be heading for the Bay of Fundy, which is a major stopover area in the fall. But these are the kinds of things that researchers are learning from this data. There was a semi-palmated sandpiper tagged in Churchill, Manitoba, that was detected at South Valley, and then the next day on the coast of Maine. And another semi-pal sandpiper was detected at South Valley, then at a state forest in Delaware, and then wound up in Brazil. So probably that was an over ocean flight that took it down there, which is pretty cool. So our Berlin station is a little different. It's mounted on the side of a dairy barn. And the brains are inside the barn. And we're able to connect to electricity there, which eliminates the need for the solar panel. Oops. So this station is a little north of downtown Berlin, between Route 16 and 110 heading northwest. So one of the first birds they detected there, other than our local rusty blackbirds, was a bird tagged in Quebec last fall on migration at that same bird observatory on the St. Lawrence. It was detected in Connecticut last fall on its way south. This spring, it was detected at 5 stations down south at the very bottom of that map, in North Carolina, Virginia, Maryland, and Delaware. And then this fall, it was detected in Quebec again, and then detected by our station in Berlin. So they really got a good sense of 3 migrations from this bird. Over in Northumberland, we have the Cape Horn station, which was installed in August. You can see this is one of the standard configurations with the 2 sections of Roan Tower. If any of you are familiar with Lancaster, Northumberland area, this station is at the south end of Cape Horn State Forest. One of its birds was one of our rusty blackbirds that is taking a western migration route. It looks like it's going to head down the Connecticut Valley. It was tagged at its nest site in Atkinson and Gilmanton Academy Grant on the 3rd of June, and in early July made a little trip down to Dixville, but was never seen or heard from again. And then on October 5th, it was picked up again, both at Crystal Mountain and South Valley, and then detected at Cape Horn on it. So it's clearly on its way south. Another bird detected at South Valley this year was a white-throated sparrow that was tagged near the Bay of Fundy in New Brunswick. Did a little zigging and zagging around up there, and then headed south and west, and was picked up on the same day, actually 10:03, not 10:33, on the New Brunswick coast, and then made it over to Dixville and Berlin on the same day. This station is not actually part of the CSWIG grant, but our team installed it, and it's in New Hampshire. This is a privately funded station in kind of affiliation with the Science Center of New Hampshire that's attached to a building in New Hampton. It was installed in August, and one of the first birds detected there was another white-throated sparrow tagged in New Brunswick came down and was detected by a couple stations in central Maine and then in New Hampton. And one of their other first stations was one of our rusty blackbirds, tagged at its nest site in Lynchtown, Maine, pretty near the New Hampshire line. And it was detected in Dixville, Berlin, and New Hampton on the same day, and then detected in Maryland. So we're hoping that we'll have some more news from that bird in the coming weeks. A hermit thrush, tagged by the same folks in New Brunswick, took a similar route to that white-throated sparrow. Didn't trigger quite the same stations in Maine, but went through New Hampton and then headed down to Delaware. The Granite Lake Headwaters station in Stoddard was the first station. Is that true? It's not quite true. It was one of the first stations installed in New Hampshire and is sponsored by the Harris Center for Conservation. It is just south of Route 9, going over from Concord to Keene. They don't seem to be in quite such a popular migration route, but they picked up a semi-palmated plover, which was tagged in Churchill, Manitoba, was then picked up by their station, then headed over to Grand and was detected by 10 different stations along the Bay of Fundy, and then headed out to sea and was detected on Sable Island in Nova Scotia and hasn't been heard from since. So I don't know if its sense of direction was a little messed up or what, but we'll see if it shows up somewhere else this fall. And one of Tatusac's pipits or horned larks was also detected in Stoddard and subsequently in northeastern Pennsylvania with an estimated minimum flight speed of 30 miles an hour. Phillips Exeter Academy has a station on the roof of the library. Whoops. And I'm going to go back to that for a minute. As you can see, this is yet another building installation with a slightly different configuration. And the receiver is located in a lab inside the library. It has power from an AC outlet. It's hooked up to Wi-Fi. So this is kind of an ideal situation because if you can get Wi-Fi, then the data is uploaded automatically. The remote stations, most of them can use cell towers to upload from. South Valley does not have that capacity, and we have to go and exchange SD cards and bring them back to upload the data on a computer. I suspect the audience is pretty familiar with where Phillips Exeter Academy is located. One of the first birds that was picked up there was a rusty blackbird tagged at Tatusac last fall, detected at 4 stations along the St. Lawrence, picked up at South Valley, and then detected in Maryland. And then this year, it was detected in northeastern Pennsylvania and then at Exeter. And there's no line on this map. I can do this. Well, that wasn't what I wanted to do. Anyway, going back to this map, the Pennsylvania and Exeter stations are located by white dots on this map, but there isn't a line between them. They picked up a Swainson's thrush that was tagged in New Brunswick on the 13th of September and hung around there for a while, left around the 26th, was picked up in Exeter on the 28th, and then proceeded in October to be detected in Delaware, Maryland, and Virginia. They also picked up one of our rusty blackbirds that was tagged at its nest site, actually in Berlin, and was detected on the coast of Maine at the Rachel Carson National Wildlife Refuge, and then down to Exeter. And another rusty blackbird that was tagged as part of a graduate student project at University of Maine was tagged at its nest site in late June in Kibbe, Maine, then detected in Norway, Maine on its way south, and subsequently in Exeter. And an American pipit, again from Tatusac, detected in New Hampshire, then Massachusetts, and eventually, after being picked up at several sites in the mid-Atlantic, was detected in Florida. So I looked at some of the minimum speeds that were calculated by Motus. A bobolink was detected at up to 62 miles an hour, sample of 8, and up to a mean of about 48.5 miles an hour. The hermit thrush, there was only one sample, 62 miles an hour. 7 samples of rusty blackbirds with a mean of 58 miles an hour. Swainson's thrush, small sample, but pretty consistent at 30 to 37. And 6 samples of white-throated sparrow with a mean of 21.5. So these are some of the things you can learn about migration using Motus that we've never, ever been able to do before with songbirds. So if any of you are interested in digging more into Motus data, you can go to the Motus website, either write down the link or you can just Google Motus and it will get you there. There is a lot of data that is open to the public. You can click on any receiving station anywhere in the world and find out what is being detected there. You can look up a species and find out what projects are being done with that species. You can pick an individual and follow its travels as we have done here. And generally spend hours following different pathways in different places. So I'm going to end here. Thank you for bearing with me, and I'll be happy to take any more questions that have arisen.
Carol, someone asked, are there any plans to put Motus stations in the Arctic breeding areas of shorebirds?
The furthest ones that I'm aware of are on the shores of Hudson Bay. It would be pretty tough in the far Arctic, I think, because the season is so short and the environment is really, really tough. You would have to use solar panels and I mean, it would be great during the summer because the sun is up all day, but you would kind of have to do temporary stations, I think, and take them down at the end of the season. That said, there's a whole series of stations along the shores of Hudson Bay near Churchill and also down at James Bay. And those are both big breeding areas for shorebirds. A question came boom through the chat while I was talking that I didn't see. So if you can grab that, Diane.
Yeah.
I think
What
it
do you
was from
think is the
Charlie Mims.
Yeah, what do you think is the next major technological advance in migration tracking?
Well, if there are satellite transmitters small enough for songbirds in my lifetime, that would be the next thing, I think. The big issue is miniaturizing batteries, and they kind of hit a plateau at about woodcock size. If that changes and you can get kind of constant GPS feed, that would be pretty amazing. But I have to say, Motus is better than I would have dreamed we'd have 10 years ago. So I'll happily take whatever we can get.
Here's a couple other questions about detection. When a bird is detected, can you tell if it's a flyover or stopover?
Yes, one of the things, well, this isn't data that's available to the public, but researchers can download all the detection data from their tagged birds. And it gives you a date and a time for each burst that a station picks up. And so some of the runs might be only 4 or 5 bursts. That would signify a bird that was a flyby, probably near the edge of the detection area for that station. You might get a stretch of 40 to 100 bursts. So that tells you that the bird probably passed more closer to the station or directly over it. You can also tell which antenna or antennas picked up the bird. So if it got picked up by just one antenna, you'd know, okay, that bird flew by the east, somewhere east of the receiving station. Or if it got picked up by multiple antennas, if it got picked up by all of them, then you'd know it flew pretty much right over the station. You can also figure out the time of the first detection and the time of the last detection. So you know how long the individual was within detection range of that station. I mentioned that one individual had spent 21 hours in the Berlin area. That's how we know that. And some of the South or Mid-Atlantic stations have had birds stay for multiple days. So then you know it's a stopover area. Did that answer the question?
I think it probably did. So it looks like birds fly over areas with stations but don't get detected. Any idea why?
Well, we don't know. We don't really know what routes they took between station A and station B when they didn't get detected. And one of the things we've been talking about is making a map that kind of cuts out all the detection areas from all the stations so that we can see what's left of the puzzle and where birds could be kind of sneaking through. Not all stations have good detection in 360 degrees. Some of them have a mountain behind them or something that kind of blocks reception. So a bird could be flying by a station in kind of a blacked out area, so to speak, and not get detected as well.
Carol, do you want to explain a little bit more about the antenna and how they're actually situated on
the
tower?
bird or on the tower? On the tower?
The towers, yeah.
The Carnegie Institute has come up with this software package that can look at the terrain in GIS for a particular station site and tell kind of what the detection area is going to be for that station. So knowing that, when they do the installation, they orient the antennas to cover the area that isn't blocked. If it's on the top of a mountain, if it was on Mount Washington, for example, you could do it in 360 degrees and get the antennas oriented typically in the 4 cardinal directions. I didn't include any configurations of antennas with their detection areas, but they have kind of an oval detection range. I can't describe it particularly well. But if you go on the Motus website and click on a station and click on the map for that station, it will show you how that station's antennas are oriented and kind of what each antenna's detection range is.
That's great. Thanks, Carol. So how is the maintenance on the towers funded once they are installed?
We're fundraising for that as we go along. We're also hoping that we can put together a full Northeastern, another CSWIG grant that will be at a much lower level, but will fund maintenance for both the Mid-Atlantic and the New England sites going forward. If we're lucky, maintenance will be minimal. One of the things they learned in the Mid-Atlantic is that porcupines like to chew on turnbuckles, and they had some issues with the turnbuckles for the guy wires supporting the stations getting gnawed almost all the way through by porcupines. Knowing that, we placed the turnbuckles higher up on the guy wires on the New England station, so hopefully we won't have that problem. I'm hoping that our stations will stand up pretty well. In the south, they have hurricanes to contend with. We have, knock on wood, have not had any moose bulling their way through our guy wires as yet. So we're hoping that that will continue. But that's been kind of the biggest thing we've been worried about with the northern stations. Those that are attached to buildings are pretty solid. Sometimes the internal equipment has some issues. We've had some problems with the charge controllers that are converting the electricity that comes from the solar panels to the batteries. But that's been our biggest problem so far.
Thanks, Carol. Mark, I'm not sure if you're up or not, but we've wrapped up the questions that were put in the Q&A or the chat. And a big thank you to Carol for this presentation. Very informative.
And a big thank you to everybody that bore with us for the duration.
Yeah, thank you, Carol. Thank you, Diane. And some of those maps that Carol was showing and was referring to, I dropped in the chat as you were talking, Carol. So a lot of folks were able to see and click on those maps and see those migrations just as you were talking about them. So thank you all for joining us tonight. As you probably saw in my email that I sent you yesterday or the day before, we have another webinar tomorrow based on, it was kind of like a pop-up webinar that we're talking about our pollinator habitat restoration here at the McLean Center at 7 p.m. You can join us then. And the next talk in this particular series will be in 2 weeks from now, featuring Scott Hecker, the Director of Bird Conservation at the International Conservation Fund of Canada. And it's called Where Migratory Birds Need Us Most. So thanks again, and we hope to see you at a webinar soon. Have a great night, everybody.
Thanks, Carol.